In this video we're going to be looking at RNA. So the first thing that we want to do is define what RNA stands for, or explain what RNA stands for. And RNA stands for ribonucleic acid. So you'll see straight away that we've got this term again, nucleic acid. So we'll know that RNA is one of our nucleic acids, and the other one being DNA, which we covered in a previous video. So it's good to refresh our memory. So we'll just go over what a nucleic acid is. It's got three components. Now the first of these is a phosphate group. And in the context of RNA, this is also going to be phosphoric acid. So that's your first component of a nucleic acid. Now the second component is a carbon sugar. It's actually a 5-carbon sugar. And in ribonucleic acid, it's going to be ribose, which is where the name comes from. And the third component is our nitrogenous bases. Nitrogenous bases. So we'll remember that there's two categories or subcategories of nitrogenous bases. And the first of these is our purines. That's our purines. And the second is our pyrimidines. Pyrimidines. Right? So, what's the difference between these? Well, our purines are a double ring structure and pyrimidines are a single ring structure. So there's 4 nitrogenous bases in RNA, much like DNA, but there's one key difference. So our purines in RNA are much like DNA. They're going to be adenine, A, and they're going to be guanine, or G. So they're the exact same as in DNA. But our pyrimidines, on the other hand, well, we've still got cytosine, which is the same as DNA, but there is uracil in RNA, which replaces thymine in DNA. So that's the key differences between RNA. The carbon sugar is ribose, and the nitrogenous base that's different is uracil, which replaces thymine in DNA. So what about the structure of RNA? Well, remember that DNA looks something like this. So we've got this, what looks like a twisted ladder structure, and in between we have these nitrogenous bases which are joining each other by weak hydrogen bonds. And in that way, DNA is double-stranded, okay? But what about RNA? Well, if we compare this to RNA, RNA looks something like this. It's actually just single-stranded, and we'll have still our nitrogenous bases, but they won't be attached to anything. They'll just be coming off a single strand. And the common thing between DNA and RNA is the basic component of each. So we'll remember that the key term that we covered in a previous video is nucleotide, which is a basic component of DNA, but it's also the basic component of RNA. So what does it consist of? Well, it consists of 1 phosphoric acid, 1 ribose, and 1 nitrogenous base. So what does this look like? So let's draw a little diagram to show this. So let's say this is our phosphate group, and then we'll take another colour, and this is our carbon sugar. We'll put R because it's RNA. And then we have a nitrogenous base. So let's say we've got uracil, just to highlight that that is one of the key differences between RNA and DNA. This is our nucleotide. So this is the basic structure of RNA. And what will happen is the backbone will be formed from a repetition of the phosphate group and carbon sugar, which is ribose. So this will just repeat again and again down the strand and form the backbone of RNA. And in between, of course, you'll have your nitrogenous bases. So let's put A for this one. So that's the basic structure of RNA. Okay, so what's the function of RNA? Put that as another heading. Function of RNA. Well, let's draw a simplified diagram. So we've got DNA. And DNA consists of the genetic material within a cell in the form of genes. And that genetic material encodes for different proteins. How does it do that? Well, how is the genetic material, how does that eventually end up as proteins within the cell? Well, that's the role of RNA. So what happens is we've got RNA. And at this stage, we've actually got some other steps in the middle. We've got gene activation. So what happens is the genes within DNA actually become available for RNA to copy them. Then we have the actual copying process, which is transcription, so the transcribing process, which is a copying process. And then we have translation, in which the RNA molecule formed from the copying process then ends up corresponding to certain amino acids, and that forms a chain into polypeptides, which end up eventually forming proteins. So that is a simplified explanation of what RNA does. And the last thing that we want to just touch on is the 4 types of RNA. So let's just draw this here. One, we have something called messenger RNA, also known as mRNA. Now, messenger RNA is the copied molecule from the DNA, from a single DNA strand, or the template strand of DNA is copied to form messenger RNA. So this is involved in the transcription process. And then we have another type of RNA called transfer RNA, also known as tRNA. Transfer RNA, or tRNA, is involved in the translation process. So that's the process of the messenger RNA eventually corresponding to different amino acids, and those amino acids being formed into a long protein, a long polypeptide chain. The third type of RNA that we have is something called ribosomal RNA. So, ribosomes, as you may know, they are a cell organelle, and their primary function is protein synthesis, so the formation of proteins. And RNA, ribosomal RNA, helps to form the components of the ribosome itself. So that's the third type of RNA. And the last type of RNA that we have is something called microRNA, or miRNA. And microRNA essentially just has a regulatory role within both transcription and translation. So it regulates the whole process of protein synthesis. So there you go, that's a quick overview of RNA.